In the modern day and age, many diseases that have killed millions over the course of human history have been eradicated or suppressed greatly. Mumps, measles, polio, and other diseases have been suppressed in developed nations, and diseases such as smallpox have been utterly eradicated, save for samples contained in laboratories in Russia and America. But one disease has remained quite deadly and quite notorious. The Ebola virus remains one of the deadliest diseases in the world, responsible for the most recent Ebola outbreak in the Democratic Republic of the Congo. But why is it that this disease has remained virulent and uncontested in the world of modern medicine? To understand why Ebola remains so deadly, we must understand its biology. Ebola comes from the virus order Mononega virales. This particular viral order has housed other deadly viruses, not just Ebola. I am referring to viruses such as the Nipah virus, bird flu, Marburg hemorrhagic fever, and rabies. But amongst all 5 of these diseases, one stands out amongst all others. It is still capable of causing great pain and death on a broad scale. Ebola, I am referring to. Ebola, like other viruses in that order, is bullet-shaped and contains a single strand of RNA. Ebola resides particularly in the family Flaviviridae and covers an entire genre of virus strains. Ebola is also a zoonotic disease, meaning that Ebola is a virus common in animals such as primates, but has now been able to infect humans. The issue is important enough that there have been issues with imported primates being infected with Ebola virus in the United States. Ebola infects a host through fluids infected with the virus coming into contact with broken skin or mucosa. This applies just as much to human transmission as much as it does to animal-to-human transmission. Upon infecting a host cell, Ebola migrates into the human liver, spleen, and lymph nodes, spreading the infection across the body. The virus remains dormant, incubating for 1 to 2 weeks. The virus soon begins attacking immune cells, leading to an overwhelming immune response. The first symptoms showing will be vomiting and fever. As the disease becomes more severe, hemorrhaging begins all over the body, including under the skin, the blood vessels having bursted due to inflammatory agents the virus releases. Shock also occurs, most likely due to the blood loss the virus causes. Eventually, organ failure sets in and the host dies. Beginning in 1976 and continuing to 2006, there were 14 different Ebola outbreaks across Africa. In addition, outbreaks have occurred in West Africa and most recently the Democratic Republic of the Congo, marking that number up to 16. Fortunately for the rest of the world, Ebola is not airborne and is considerably less infectious than, for instance, the flu. Unfortunately, the Ebola virus is extremely lethal. The deadliest variant of Ebola virus, the Zaire Ebola virus strain, has a case fatality rate of roughly 83%, but in certain outbreaks it has reached as high as 90%. Compare this to the Spanish flu, which had a 10 to 20% fatality rate. Furthermore, the Ebola virus is readily transmissible enough that nurses and doctors in outbreak zones wearing proper safety equipment have been infected and died from the virus. With this in mind, along with the large number of past and ongoing outbreaks, finding a medical solution to the virus is one of the medical world's highest priorities. Currently, combating the virus has remained in the realm of treatment. Treatments include infusion of fluids, electrolytes into the body, oxygen therapy, medication to alleviate symptoms, and treating other diseases or infections that may occur due to the patient's reduced immune system. The Ebola virus is capable of quick mutation, and there are various strains already in existence. The 2014 West African Ebola strain was a mutation of the already lethal Zaire Ebola virus strain, for instance. The many outbreaks, past and current, show that it is hard to contain the virus, especially given the circumstances in Africa, specifically the lack of established medical infrastructure and sanitation. With that being said, research has yielded new possible methods for combating Ebola. At the U.S. Army Medical Research Institute for Infectious Diseases, Fort Detrick, Maryland, scientists had developed an experimental vaccine that allowed macaques to survive an Ebola infection. When a macaque was injected with the vaccine, a day after being infected with the Ebola virus, the macaque survived. However, the control group did not. Other research has also been conducted in using the blood of surviving Ebola patients to form a serum to give to infected individuals. The latter treatment was approved by the World Health Organization in 2014. In 2015, the WHO reported that an Ebola trial vaccine was extremely promising. However, at the time there were 15 different vaccines in development, though, in addition to that fact, there were 2 in late-stage development, probably indicating why it was stated that it was extremely promising. But despite not being approved by a regulatory committee, there is enough faith in the vaccine that approximately 300,000 doses are stockpiled. In the most recent outbreak in the Congo, Uganda injected the vaccine in more than 8,000 individuals along the Congo border to prevent the spread of the virus into the country. That being said, despite the preventative measures that other countries have taken, there is still an ongoing outbreak in the Democratic Republic of the Congo, and there is yet to be a mass vaccination program there, either indicating lack of resources, lack of interest, or lack of an effective vaccine that can cover the whole area, and the high mutation rate of a virus. The high mutation rate of the Ebola virus means that a vaccine may prove ineffective in a short time, meaning that an Ebola vaccine for places like the Congo or West Africa might have to deal with something similar to flu vaccines, which must be developed every year. In the world today, despite advances in medical technology, the Ebola virus by its very nature poses a significant challenge for antiviral treatments, vaccines, etc. The Ebola virus, though capable of unusual feats of infectivity, is not airborne, currently, and does not infect a large number of people each year, meaning that the pool of researchable patients is small to begin with. Ebola virus strains must also be kept in level 4 biosafety labs. In layman's terms, the maximum security for a biological research laboratory. For this reason, the vast majority of scientists across the world who do not have access to military-grade biological research laboratories are not able to study the virus safely. You can't really keep Ebola in a hospital safely. At the same time, smallpox was being eradicated, for instance. Ebola was killing people by the thousands in Zaire, what is now known as the Democratic Republic of the Congo. Additionally, the Ebola virus has the ability to mutate rapidly, evidenced by the West African Ebola outbreak of 2014, which was a mutated strain of the Zaire strain of the Ebola virus, meaning that it was more infectious, but slightly less lethal. Because of the ability to mutate rapidly, Ebola virus is capable of adapting to known vaccines and treatments, becoming more infectious or more lethal, but in rather universal terms, resistant to the treatments and vaccines we may use. Similar to how bacteria have been adapting to antibiotics recently. Compounding this is the issue that antiviral medications suffer a severe lack of development compared to antibiotic medications. The result is a virus hard to study widely and harder to treat, and capable of retreating into the jungles from which it came every time it is contained, only to come out once more, though different within genetically, but bleeding its victims all the same. Thousands may die before a vaccine is found, a vaccine that could be universally used. But even then, it is unlikely to say that this would work.